Alright, so chapter 12 concerns the innate immune system. It's kind of an overview for the immune system in general, alright, because we'll talk about how the innate immune system and the adaptive immune system interact. Alright, and later on we'll talk about disorders in chapter 14. Alright, so but first, we're going to focus our attention on the innate immune system. Alright, so thinking about immunological barriers. Alright, you have primary, secondary, and tertiary barriers. Alright, so your primary barriers, alright, these are going to be, for example, physical barriers. Alright, so think about skin. The skin, our skin cells, are constantly shed. Alright, so that's a physical barrier. Alright, it's also coated in a nice waxy secretion. Alright, it's keratinized. Alright. We have the physical movement of microorganisms from the throat. Alright, so in this case, your pharynx, alright, as it's entering your respiratory tract, you have ciliated columnar epithelial cells that sweep away trapped microorganisms or any type of antigen in that case. You also have a mucus that traps these organisms as well. Alright, you have physical barriers like the fact that you blink, or sneezing, or you have hairs within your nostrils. Right, they're physical barriers. They're there to prevent the entry of microorganisms. All right, you also have chemical barriers. All right, so thinking about, for instance, tears. All right, your tears contain many chemicals, peptides, along with things like lysozyme that break down the cell wall of bacterial cells. You have sebaceous glands that secrete oils that are highly acidic. You have your highly acidic conditions in the stomach. You have your alkaline conditions within the small intestine. You have the presence of bile salts in the small intestine. All right, you have saliva, which contains lysozyme and other degradative enzymes. And then you also have your own genetic barrier. All right. In this case, the genes that comprise the various cells of your immune system, right, which is specifically unique to you as an individual, from which you inherited from your parents, is going to basically predetermine, outline your most basic immune defenses. And then you also have a microbial barrier, right, both on and within your body. All right, so you have your own microbiome, your own personal microbiome, all right, that serves the function of serving as a physical barrier for the attachment and entry of microorganisms, all right, as well as a way to basically prime your immune system as an infant. Right? Your immune system begins to develop upon exposure to microorganisms that you come into contact with through either it be vaginal delivery or cesarean delivery, organisms you come into contact through touch with other individuals, the environment you're in. And all this is used to train your immune system to ultimately differentiate the cells of your body from other types of cells. All right, that shouldn't be there. All right, secondary barriers. Secondary barriers are going to be things like your leukocytes. All right, so you have your leukocytes, like your macrophages, all right, your neutrophils, and your dendritic cells. All right, all these guys are examples of phagocytic cells. All right, so their job is to engulf foreign antigens, ultimately destroy them, and also present those foreign antigens to T cells. You have process of inflammation, so you have a inflammatory response, all right, which is there to not only attract immune cells to a site of injury, all right, is there to promote the response of clearing away any foreign invaders. It primes the immune system, put on high alert, as well as you have the involvement of other factors like complement, and you have factors like certain chemicals that we call pyrogens that stimulate fever. All right, and the fever is also involved as a secondary barrier. Your tertiary barrier is going to be your lymphocytes. All right, so in this case, your lymphocytes are going to comprise of your B cells and your T cells. All right, so in this case, we're talking about humoral and cell-mediated immunity in terms of your T cells. Now we're going to talk about each of these things independently on their own in terms of primary, secondary, tertiary barriers, but it's important that you come to realize the interconnectedness of these three lines of defense and how they're all interrelated. All right, so this is giving you an example of that mucociliary escalator. All right, so here looking at your trachea, all right, you have your pseudocolumnar epithelial cells. All right, so you can see here they're ciliated. Gives you a little blow-up image here of a cilia, and it's their job to basically remove any pathogens, any antigens that get trapped in this mucus. All right, you have these little goblet cells here. Their job, the goblet cells, is to basically secrete this mucus. You have your defensins, you have lysozyme, all right, so you have antimicrobial peptides, all right, that function in destroying microorganisms. So lysozyme targets the N-acetylmuramic and acetylglucosamine, the sugar components, the glycan component of peptidoglycan. All right, so it basically severs this glycosidic linkage between your sugar residues. All right, basically separating, cleaving cell wall. All right, you have defensins, other types of antimicrobial peptides that intercalate into the membrane of bacterial cells. And so by intercalating into the membrane, what they do is they disrupt this barrier by creating this channel, by creating this pore. All right, so the cell can no longer regulate its own osmotic potential. All right, so cellular constituents leave and others come in. So it's highly susceptible to lysis at that point in time. All right. You also have within your gut, you basically have this collection of lymphatic tissue. All right. You have these collection of lymph nodes, all right, within the intestines that we refer to as Peyer's patches. The whole function of these Peyer's patches is that they serve as a localized site of immune cells, so B cells, macrophages, neutrophils. So it's a fairly quick response in terms of the mucosal immunity of the gut. All right, so looking at these various scenarios and determining whether or not they are examples of physical, chemical, or genetic barriers. So hydrochloric acid stomach. Well, this is a chemical barrier. All right, so in this case, the low pH of the stomach is going to, in this case, kill most of your microorganisms. All right. In this case, these organisms cannot function at that low a pH in terms of their physiological responses, denaturization of proteins, damage to proteins, DNA, lipids. All right, sloughing the skin. All right, so this is an example of a physical barrier. Lysozyme and tears saliva, example of chemical barrier. Mutation in a gene for complement proteins. It's genetic. Ciliary escalator. Physical. Thinking about the actual physical movement of substances through your digestive tract. Thinking about the process of defecation or urination. All right, both of these are also examples of physical barriers. All right, so when you're looking at the immune system, all right, we're looking at what its ultimate role is. What is its job? All right, the job of the immune system is to differentiate, discern between our own host cells that are healthy versus our own host cells that are unhealthy, infected, and or other foreign antigens. All right, so here you have a white blood cell that's coming into contact with cells of our body, all right, that are presenting normal antigens, normal self-antigens. All right, so here, this presentation involves what we call MHC1 receptors. All right, so these MHC1 receptors are found on all of your nucleated cells of the body. All right, so these MHC1 receptors function in presenting antigens to our immune cells. All right, so if these are self-antigens and they're normal, they're not modified in some way, or there's not an abnormal concentration of certain receptors or certain cellular components being presented, then our immune system, all right, says everything's a-okay. All right, there's no immunological response. Okay. Now, when a white blood cell comes into contact with some foreign antigen, all right, so it could be a microbial cell, all right, these little receptors on the surface of our immune cells, all right, specifically our leukocytes, are referred to as pathogen recognition receptors, or PRRs. And their job is to very specifically bind to certain microbial antigens. All right, so pathogen associated molecular patterns, or PAMPs for short. All right, so once they detect these foreign cells, they're targeted for destruction, all right, and elimination. All right, so that's our leukocytes. Our lymphocytes also function in a similar manner. All right, our lymphocytes, like our T cells, all right, they have T cell receptors that bind to specific antigens being presented to them. All right, so they'll bind to self antigens being presented to them and bind to non-self antigens. All right, so if it's self antigens, the T cells are fine; they're a-okay. All right, they don't start proliferating. There's no cell-mediated response. But if there's non-self antigens that are presented to the T cell, all right, they're going to receive a secondary signal. All right, letting them know that they need to proliferate and begin differentiating. All right, and later on we'll talk about specifically different types of cell-mediated responses. All right, just know that you can have both your leukocytic cells and your lymphocytic cells that are being stimulated by the same type of antigens, the same antigens in this case. All right, so the lymphatic system. All right, so the lymphatic system is highly integrated with our cardiovascular system. All right, so here the function of the lymphatic system is to drain extracellular fluid from tissues of the body. All right, so this extracellular fluid is leaking from the vascular outside into surrounding tissues, and then is eventually going to be forced into the lymphatic vasculature. So you have these lymph capillaries. All right, so you have fluid in surrounding tissues, extracellular fluid, that enters lymphatic capillaries. All right, these lymphatic capillaries transport the lymph through lymph nodes. All right, so it's very similar to how our vessels transport our deoxygenated blood back to the heart. All right, for oxygenation. All right, so the movement of lymph through the body requires both the movement of voluntary skeletal muscles and the involuntary movement of smooth muscle throughout the body. This is muscle contractions that allow the pumping or movement of lymph. All right, now the lymph, as it makes its way back to the ultimately back into systemic pulmonary, whether it be pulmonary or cardiovascular circulation. The lymph is going to pass through lymph nodes. Now, as this lymph passes through the lymph nodes, it's going to come into contact with immune cells, all right, within the lymph nodes. All right, so it's going to come into contact with macrophages. It's going to come into contact with B cells, as well as T cells. All right, so this is where you have a mixing and mingling of your innate immune cells and your lymphocytes involved in the adaptive immune system. And eventually the fluid within the lymphatic vasculature is going to be dumped into veins of the heart. All right, through the jugular veins, through a lymphatic duct into the heart. All right, so looking at our lymph nodes a little bit more closely. All right, so the lymph as it enters the lymph node is referred to as afferent lymph. All right, and then efferent lymph is actually leaving our lymph nodes. All right, as the lymph passes through our lymph nodes, is going to come into contact with B cells and macrophages and T cells, dendritic cells. And your lymph composition is very similar to the composition of fluid found within your capillaries and your veins, except for the fact that you're not going to find plasma proteins. All right, they're just too big. They're not going to enter the lymph. And you also have the transport of fat through the lymph as well. All right, but this is how our immune cells, like our phagocytic cells, they come into contact with an antigen, they engulf it, and they make their way by entering the lymphatic vasculature. They make their way to a lymph node, typically localized near the site of injury or infection. And this is where they present antigens to T and B cells. This is within the lymph nodes. Alright, so your B and T cells. Alright, so both your B and T cells originally are going to originate from the bone marrow. Alright, so you have undifferentiated stem cells. Alright, they're going to come from the old sac of a developing embryo. You're going to have these stem cells that are hematopoietic. Alright, so they can differentiate into all different types of cells. Alright, so these hematopoietic stem cells are going to be within the bone marrow, and these cells are going to leave the bone marrow, differentiate, in this case, into B cells. They're B cells, they'll leave the bone marrow and travel to the lymph nodes. Alright, so B cells come from bone marrow. T cells also come from the bone marrow, but they migrate to the thymus. Alright, so in this case, here the B cell is naive. Here the T cell is also naive from the thymus. These will enter the lymph nodes. Alright, so these are your little B and T cells that have made their way through all the appropriate screening processes. Alright, we'll talk about positive and negative B and T cell selection later on. Alright, but once they've made their way from the bone marrow or thymus, they're going to also migrate to lymph nodes, because you have a collection of lymphatic cells within your lymph nodes. Alright, and this is where they're going to become primed. Is when they come into contact with an antigen, all right, specifically an antigen that's going to be presented by an antigen presenting cell. All right, so in this case, your macrophages, all right, your dendritic cells, as well as your neutrophils. Okay. Now, you also have a collection of lymphatic tissue in different areas of the body. All right, so here you have, within the lungs, you have mucosal associated lymphoid tissue. All right, also referred to as MALT. All right, you have a collection of lymphatic tissue within the gut. All right, so you have these, what we call Peyer's patches. All right, these Peyer's patches serve as a collection of T and B cells, some mucosal immunity. All right, so MALT, mucosal associated lymphoid tissue. You also have GALT. All right, you have gut associated lymphoid tissue. All right, so these hematopoietic stem cells in the bone marrow give rise to stem cells for all other types of cells, all right, such as your erythroblasts become your red blood cells. You have your megakaryoblast give rise to your platelets. You have your myeloblast, which give rise to ultimately your leukocytes. All right, so you have your neutrophils, your basophils, your eosinophils, and your monoblast give rise to monocytes. All right. You have other stem cells, all right, lymphoid stem cells that give rise to T and B cells. All right, so looking at each of these specific types of cells. All right, so in this case, your basophils and your mast cells are mainly going to be involved in allergic responses. You may see an increase in eosinophils as well. All right, your basophils and mast cells contain chemical modulators that basically trigger the inflammatory response and subsequently the symptoms that you experience during an allergic response and coming into contact with an antigen that's triggering the allergic response, something called an allergen. All right, so your eosinophils. All right, so a higher than normal titer of eosinophils within the blood is going to indicate usually some type of parasitic infection or fungal infection. All right, your neutrophils, your macrophages and dendritic cells all function as phagocytic cells. They're also antigen presenting cells. All right, now your monocytes remain within the blood. Once your monocytes move into the tissues, they're now referred to as macrophages. All right, your T and B cells. So we're talking about B cells, humoral immunity, production of antibodies. T cells are cell-mediated immunity. And you also have a group of cells referred to as natural killer cells. Alright, so lymphatic fluid contains all the following except what? White blood cells. Yep. Extracellular fluid. Check. Infectious agents. Possibly. Cellular debris. Possibly. Red blood cells. No. Alright, so looking at pathogen associated molecular patterns, and then pathogen recognition receptors. Alright, so these are PRRs and these are our PAMPs. Alright, so these pathogen associated molecular patterns are going to be typically biological molecules. Alright, typically it's going to be protein, carbohydrates, that not always. You can also have lipids and nucleic acids, but usually it's protein and carbohydrates that are triggering the response. Alright, and these are specific structures that you would find on microbial cells and not find on host eukaryotic cells or within them either. Alright, so things like lipopolysaccharide, which you would find in gram-negatives, flagellin. Alright, so bacterial flagellin and eukaryotic flagellin are two totally different types of molecules. Alright, peptide glycan you're not going to find in a eukaryotic human cell. Alright, you also have microbial DNA, you have viral nucleic acids. Alright, so any type of structure that is not typically found in a host cell is going to trigger an immune response. Okay, so there are specific pathogen recognition receptors that we refer to as toll-like receptors. Alright, or TLRs. And these toll-like receptors are very specific. Alright, so even though your innate immune system is usually described as being a non-specific response, it's still specific. Alright, because it's interacting with and targeting specific molecular structures. Alright, so these toll-like receptors are very specific to the types of molecular structures that they bind to and identify. Alright, so toll-like receptor 4 binds to LPS, toll-like receptor 5 binds to flagellin, toll-like receptor 2 binds to peptide glycan. Alright, so they're very specific pathogen recognition receptors. They recognize very particular molecular structures. Now, the overall response of coming into contact with these structures is a generalized response. All right, it's going to be more or less the same response every time. in terms of the innate immune response. Alright, so looking at the process of phagocytosis. Alright, so you should be able to outline, alright, and describe the steps that are involved in the phagocytic process. Alright, so first you have the attraction, alright, of your phagocytic cells to your microorganisms. Alright, so in this case you have the migration of your phagocytic cells to the site of injury or a site of infection. Alright, so this process is referred to as chemotaxis. Alright, so there are basically these little molecular breadcrumbs, alright, these signals that cells that have been injured or other phagocytic cells are releasing that are most concentrated at the site of injury or infection and then diffuse out. Alright, so the phagocytic cells use this as a way to migrate and find the site of injury. Alright, they move towards the higher concentration of these chemical signals. Alright, next, alright, you're going to have adhesion. Alright, basically the attachment of these microbial cells to your phagocytic cells. Alright, so this is going to be an interaction between the PAMPs and the pathogen recognition receptors. Alright, next, you're going to have the actual engulfment, the formation of this pseudopodia, right, around, it envelops and encases your microbial cell, and then it's engulfed. And so at that time, that little vesicle is referred to as a phagosome. All right. Now, next, you're going to have the fusion of your phagosome with a lysosome. All right, so you have the formation of a phagolysosome. All right, and this phagolysosome, it contains, all right, reactive oxygen species. It contains nitrogen oxygen species. It contains hydrolytic enzymes. Alright, so things like lipases and proteases and you have enzymes that degrade carbohydrates. You have DNases. You have RNases. Alright, all these are ultimately going to destroy that microbial cell. Alright, so you have destruction of the microbial cell. Alright, all these little cellular components get chopped into little bits. And then you have the elimination of those digested materials outside the cell. Now, some of those components are going to go through a process in which they're going to be presented. Antigen presentation. Alright, so antigen presentation on MH MHC1 and MHC2 complexes. All right, we'll talk about those a little bit later on in chapter 13. All right, but just know that you have antigen presentation. These foreign antigens that have been digested, those little bits, are going to be expressed on the surface of your phagocytic cells, your antigen presenting cells. All right, and the function of these antigen presenting cells is that they present these antigens to T cells. All right, so this is a connection between your innate and your adaptive immune response. You have antigen presentation from innate to the adaptive. All right, so looking at the steps, all right, for phagocytosis. All right, so what's the first step? Well, you're going to have chemotaxis by a phagocytic cell. All right, you're going to have adhesion. You're going to have engulfment. You have phagosome formation, phagolysosome formation. You're going to have killing and destruction, and then release of debris and antigen presentation. All right, so one of the next innate immune responses is the inflammatory immune response. All right, so there are 5 signs associated with the inflammatory immune response. All right, so you have rubor, you have calor, you have tumor, you have dolor, and then you have loss of function. All right, so the rubor is referring to red. All right, so the fact that you see redness, all right, so the redness is indicative of increased circulation to the site of injury or infection. In this case, you have increased vasodilation. All right, so increased blood flow to that area, so that's why it appears red. All right, the calor. All right, the calor is indicating warmth. All right, so along with this, you have the movement of blood from the core of the body to the extremities, typically. All right, in this case. So the core of the body, the temperature is a little bit warmer than the extremities, so in this case, the blood moving to the extremities is much warmer, so you feel this area of redness along with this warmth. So it's the heat that's being given off by the blood. All right. You have tumor, which is indicative of swelling, right, or edema. All right. So along with this, the vasculature near the site of injury, you have increased vascular permeability. All right. So that means that the fluid plasma within the vasculature leaks into the underlying tissues. All right, so increased extracellular fluid, basically. And so it causes that area to swell. The dolor, all right, the dolor indicates pain. All right, so the nerve endings near the site of injury or infection are going to be stimulated. All right, so your pain receptors are triggered. All right, and as a result of this inflammatory process, you can lose loss of function to that particular area, whether it be a digit, it be an appendage, and a limb. All right, there's going to be a loss of function due to the swelling. Now, in this inflammatory response, all right, you have a couple of immunological mediators that are going to play important roles. All right, now I want you to become familiar with the function of these particular immunological mediators. All right, the ones that I'm holding you responsible for are the ones that have been starred. All right, so here you have your non-specific mediators of inflammation. All right, so tumor necrosis factor. All right, you have interferons. You have interleukin 1. All right, interleukin 1's really involved in stimulating a fever. All right, tumor necrosis factor is involved in chemotaxis and stimulating phagocytosis. It's also functioning as an endogenous pyrogen. All right, so this also triggers a fever. All right, you're going to see a lot of crossover between certain immunological mediators and what they do and what they trigger. All right, your interferons are involved in typically virally infected cells. Basically, they serve as a warning signal that a cell has been infected by a virus, and that signal triggers other nearby cells to basically be on the alert, all right, so as to impede the entry of those particular viruses, all right, to amp up their own cellular defenses. All right, you have vasoactive mediators. All right, so in this case, you have your mast cells, your basophils, sorry, that basically trigger your vasculature, like histamine, to dilate. All right, you have increased vascular permeability, increased fluid movement from vasculature and surrounding tissues, increased mucus production. Well, all of these are examples of what you would typically see in not only an allergic response, but as well as an allergic response to an antigen. All right, so you have things like bradykinin are involved in pain. They're also involved in vascular permeability, triggered smooth muscle contraction, involved in allergic reactions. And then you have the prostaglandins. All right, prostaglandins are involved in inflammation and pain, triggering dilation or constriction of vasculature. Leukotrienes. All right, so histamine is not the only trigger for an allergic response. You also have leukotrienes that also play a role in stimulating the contraction of smooth muscle, which you would find within the throat. All right, so your pharynx, your trachea, and so this causes your symptoms such as more severe manifestations of allergies like anaphylaxis. All right, it's a constriction of the airways through the contraction of your smooth muscle cells. And you also have platelet activating factor, all right, which causes basically platelets to aggregate. All right, so basically to kind of ward off and close off the site of injury or infection. All right, so looking at this inflammatory response. All right, so you have some site of injury, for instance, all right, and pathogens are going to enter this wound. All right, so this injured tissue and macrophages that are already present nearby in the tissue are going to begin to, in this case, the macrophages engulf foreign cells, foreign bodies, and your injured cells, along with the macrophages, secrete these chemokines. And these chemokines are those basically the little breadcrumbs that are of highest concentration at the site of injury. And then as they diffuse out, right, they become in smaller quantities, concentration decreases. All right, so your immune cells migrate towards the site of injury following these breadcrumbs, following these chemokines, right, from an area of low to high concentration. All right, you have in this case, you have your mast cells. All right, you have your basophils that begin to release chemical modulators like your histamine. They cause this vasculature to become increasingly leaky. All right, increased vascular permeability, so that now the fluid in the vascular leaks into the surrounding tissues. All right, so you have swelling, you have blood flow that's diverted to the site of injury. All right, you have a pain response. All right, so that's going to be triggering all four of those signs of inflammation. All right, you're going to have your neutrophils that eventually migrate to the site of injury. Now, these neutrophils and these monocytes, the reason they can move through the vasculature is because actually at the site of injury, you're actually going to have vasoconstriction. All right, so in this case, the vasculature shrinks so that the blood flow actually slows down enough so that your immune cells, like your monocytes and your neutrophils, can actually adhere to the surface of the endothelial cells. All right, so they're going to go through this process of rolling. They attach to certain receptors along the endothelial cells. They adhere to the surface of the endothelial cells, and they're going to begin to kind of spread out, and then they're going to move from the vasculature into the surrounding tissue. There's a process of extravasation, or what we refer to as diapedesis. All right, so the process of diapedesis. All right, so once these immune cells, these leukocytes, migrate into the tissues, they begin engulfing all the foreign cells, bacteria, and begin to go through this process of phagocytosis and the elimination of whatever's left over through the digestive process of these foreign cells, as well as you have, once these guys have actually come into contact with the immune cells, they're going to actually re-enter the vasculature, and they're going to move onward to your lymph nodes, all right, where you're going to have a process of antigen presentation. All right, because you're going to have those antigens that are presented on MHC complexes to your T cells. Eventually, you're going to have the formation of platelets around the site of injury. You're going to have a process of wound healing. All right, so in this case, looking at a fever, all right, which kind of coincides with this process of inflammation. All right, you have what we call pyrogens. They can be endogenous pyrogens. All right, so your endogenous pyrogens are going to be basically the products of your phagocytic cells engulfing your foreign antigens. All right, these products that are released and presented function as endogenous pyrogens. You have chemical mediators like interleukin 1 and tumor necrosis factor alpha, which function as endogenous pyrogens. But then you also have pyrogens that are exogenous. Alright, so these are going to be basically foreign antigens. Alright, in this case, it could be something like, for instance, LPS is a pyrogen. Alright, so these are going to be molecules that you would find on microbial cells that stimulate fever. Alright, so what are the benefits of fever? Alright, well, the increased core temperature of the body. Alright, so this triggers the hypothalamus to adjust and alter the core set temperature of the body. In this case, increased body temperature, a mild grade fever, 100 degrees, is going to disrupt the ability of that microbial organism to replicate. Alright, so tying this back into the growth requirements of microorganisms. They function best at a set range of temperatures. By altering that temperature range, by altering the temperature of the host, alright, it's going to be outside the range of that microbial cell in terms of replication. So you slow down the growth. Alright, you also increase the sequestering of iron from the blood. Alright, so iron is one of those particular factors that microbial cells need in terms of their proteins and functioning appropriately. And so by sequestering those by the body, alright, you prevent access to iron, or free access of iron to your microbial cells. You also stimulate your immune cells by increasing temperature. This basically primes them for a much more heightened response. Alright, so it speeds up the production of immune cells, it speeds up the process of phagocytosis. Alright, now if a fever gets out of hand, so something above 104 degrees, 105, 106, alright, you're reaching lethal levels, right, where you're basically essentially boiling the brain. Alright, you're basically not only affecting microbial cells, you're also beginning to affect host cells of the body, and you start disrupting host cell proteins, host cell physiology. Alright, because the cells of our body function within also a pretty narrow range of temperature. Alright, so along with a fever, you can have, for instance, tachycardia, so increased heart rate. Alright, you can have tachypnea, so increased respiratory rate. You can also have the increased risk of developing a seizure. Alright, so interferons. Alright, so you have a virally infected cell. Alright, this virally infected cell is going to detect foreign viral nucleic acids, alright, through toll-like receptors. in the nucleus and nuclear membrane. And you have interferon genes within the host cell DNA that's actually triggered. In this case, interferon synthesized is released by the infected cell, and it binds to nearby neighboring cells. And it triggers that particular cell to then begin secreting antiviral proteins, so that it blocks the viral replication. If a virus happens to enter that cell, it already knows that a virus is nearby, so it's already on the defense.
When a cell is infected by a virus, the virus enters the cell and produces structures that are not found in uninfected cells. The presence of this viral material signals the cell to produce interferon. The interferon moves out of the cell and attaches to receptors on nearby cells of the same type. The cell that produces the interferon is unable to save itself. The virus replicates in this cell and then moves out to infect nearby cells. The nearby cell that already has interferon bound to its surface responds in several ways, including production of enzymes that degrade messenger RNA and prevent protein synthesis. Thus, a virus can attach and enter the cell, but completion of the viral replication cycle is prevented.
Alright, so complement. Now, your book talks about two other types of complement pathways. Alright, so it talks about an alternative complement pathway. It talks about a lectin-mediated pathway. Really, I'm going to have you focus mainly on the classical complement pathway. Alright, so here, alright, this is a antibody-dependent pathway. Alright, so in this case, you have antibodies that coat the surface of your foreign cells. You have a protein complex of complement factors. Alright, C1 proteins that adhere to the antibodies. Alright, so here, if you look at antibody structure, alright, this little Y-shaped structure. Alright. You have this region here that's referred to as the constant region. This region up here and here is referred to as the variable regions. Alright, so the constant region is always going to stay the same, in the sense that it signifies what type of antibody you're dealing with, whether it's IgG or it's IgM, or we'll talk about different antibodies later, but it signifies what type of antibody it is. Alright, the variable region is what actually binds to your foreign cells. Alright, so it binds to the antigen. It's an antigen binding region of the antibody. So here your complement factor actually binds to your constant region of your antibody. Alright, so upon binding, you have a series of other complement proteins that bind to this protein complex, alright, and begin attaching to the surface of the microbial cell. Alright, so you have C2b, C4b, C3b, C5. All right, typically the B subscript is for binding, all right, in terms of attachment to the cell surface, and the A is for away. All right, these function as it says, inflammatory mediators. All right, so they trigger the inflammatory response. All right, so there's a lot of crosstalk between the inflammatory immune response, phagocytosis, complement, and humoral immunity. All right, so you have these series of complement factors that bind to the host cell, but the microbial cell surface. All right, so then you have C9. All right, so you have C5, 7, 6, 8, and then C9. C9 basically forms this complex that basically encircles and encases these proteins, and it basically forms a pore. All right, so this pore, or what we refer to as a membrane attack complex, pokes a hole in the membrane of the cell, so that now the cell can no longer regulate its osmotic potential, so it's more highly susceptible to lysis. All right, so here you can see the products of that. You can see all these different holes in the surface of the cell. All right, so complement, besides actually targeting and destroying microbial cells through this membrane attack complex, it also serves in inflammation. All right, so it triggers mast cells to secrete histamine, which promotes the migration of immune cells to the site of injury. It allows for the movement. Right, so these are also chemotactic factors, all right, that also tell your neutrophils and your macrophages where to go, right, in the body. And you have also phagocytosis, all right, so the binding of complement to the surface of a phagocytic cell. All right, you have complement receptors on your phagocytic cells. You have complement that binds to the surface of the microbial cell, and you can have the engulfment of a microbial cell through interaction with complement receptor proteins and complement factors, along with antibodies and antibody receptors. All right, so complement's involved in many different things: inflammation, membrane attack complexes, phagocytosis, chemotaxis, as well as this process of opsonization. All right, so complement factors coat the surface of your microbial cells, and so they make them more recognizable and seen. And I guess, for lack of better terminology, all right, they make them more yummy to your phagocytic cells.
Complement consists of a group of serum proteins that activates inflammation, destroys cells, and participates in opsonization. Complement can be activated by a number of different foreign molecules. The complement proteins respond in a sequential manner, producing a cascade of reactions. The major components are C1 through C9, named in the order that they were discovered, not in the order in which they function. The complement cascade can be activated by the classical pathway or by the alternative pathway. In the classical pathway, C1 becomes activated when it binds to an antigen-antibody complex. The activated C1 then cleaves C2 into C2A and C2B, and C4 into C4A and C4B. C2b and C4b combine to form a protease called C3 convertase. C3 convertase then cleaves C3 into C3a and C3b. In the alternative pathway, antigens such as endotoxin, polysaccharides, or cell wall components react with C3b. Small amounts of C3a and C3b are constantly being formed from C3, but without activation they are soon destroyed. C3b reacts with the proteins factor B, factor D, and properdin to form a complex called C3 convertase, which cleaves C3 into C3a and C3b. Both of these pathways of complement activation follow the same sequence after cleavage of C3. C3a is involved in stimulating inflammation. C3b reacts with other complement components to form C3 convertase, which forms more C3a and C3b. C3b also attaches to the surfaces of microorganisms. Phagocytes have a binding site for C3b. Therefore, microorganisms with C3b bound to their surfaces are more susceptible to phagocytosis. Coating of bacteria to make them more susceptible to phagocytosis is called opsonization. Addition of properdin to C3 convertase results in formation of C5 convertase, which cleaves C5 into C5a and C5b. C5a enhances inflammation and acts as a chemoattractant for phagocytes. C5b reacts with other complement components including C6, C7, C8, and C9 to form a membrane attack complex. This structure forms a hole in the cell membrane and causes cells to lyse.
Alright, so this is a nice little summary slide for the innate immune system. Alright, so looking at your physical barriers, your phagocytes. Alright, so you have, later on we'll talk about natural killer cells. Alright, a little bit later on. Your inflammatory response, fever, interferons, and complement.